Okay, so good evening. First of all, I would like to thank the scientific committee for giving me the opportunity to present you my work today. So in this work, we established a new role of GPRC5A in the keratinocyte mechanotransduction. Before to start, I would like to introduce some key notion, which is about epidermis homeostasis. So the homeostasis epidermis, the epidermis homeostasis depend on several characteristics. This including the balance between the proliferation and the differentiation, as well as the well organization of the epidermal cells through the intercellular junction. Moreover, the epidermis homeostasis is closely linked to the mechanical properties of its underlying tissue, which is the dermis. It has been described that the dermis mechanical properties is around 5 to 10 kilopascal. So it has been also described that the extracellular matrix stiffness affects many functions processes. So in skin, the stiffness promotes the proliferation, the migration, and also the wound healing processes. But whereas the stiffness inhibits the differentiation and the adherent junctions formation, and at the end, alter the skin barriers. Concerning the wound healing, it has been shown that the stiffness is strongly associated with a better skin regeneration. In fact, Goffin showed that the wound granulation tissue is increased during the wound healing from 18 to 30 kilopascal. Moreover, injection of the hydrogel with different stiffness in the wounded skin in mouse show that an optimal stiffness is necessary to improve the wound healing process by promoting fibroblast transformation, wound healing, wound closure, and the keratinocyte proliferation and migration. Moreover, many studies have shown that the keratinocytes are able to sense and respond to the mechanical changes. In our laboratory, we also demonstrated that the human keratinocyte also responds to the stiffness substrate. To do that, we use the polyacrylamide hydrogel, in which the concentration of the acrylamide and the bisacrylamides are changed in order to obtain different rigidities. And in order to promote the keratinocyte adhesion, the gel surface was functionalized with the type 1 collagen. Using 3 different rigidities that we know that we call soft, medium, and rigid, compared to the glass cover slip, we show that on the soft hydrogel, the keratinocyte cell shape remain round and no clear focal adhesion points was observed. This morphology is associated with an inhibition of keratinocyte proliferation. When we observe keratinocyte morphology on the medium, the rigid, and the glass, we observe that the cells were more spread with a well-defined focal adhesion point. This morphology is associated with a keratinocyte proliferation. Moreover, we also demonstrated that the cells in the soft didn't proliferate, but when these cells were replaced on the plastic surface, a favorable condition to proliferate, the keratinocytes are able to proliferate again. In the same way, we also measure keratinocyte colony formation efficiency. We observe that during the 2nd generation, the keratinocyte from a soft hydrogel decrease the colony formation compared to the keratinocyte from glass. This observation was reversed during the third generation. So our question is by which mechanism the keratinocytes are able to re-engage their proliferation. So our objective is to identify the actor that involved by the keratinocyte mechanosensing in response to the stiffness. So we perform RNA sequencing where we compare the profile expression of the keratinocyte culture on the glass cover slip, on the hydrogel with a soft rigidities, and the condition where the cells were replate that we named the soft glass condition. This ambient approach reveals a strong signature of the glass condition compared to the soft and the softer glass. When we analyze the gene profile expression, we observe many genes are differentially expressed. Despite a strong signature of the glass condition, when we compare the softer glass versus glass condition, we still find 43 overexpressed and 62 repressed genes. Focus on making of these genes potential candidates to in keratinocyte mechanosensing. Focus on the overexpressed gene, we identify a potential candidate, which is GPRC5A, for G protein coupled receptor class C, family 5, member A. We choose GPRC5A because of its high expression during the transition, and because GPRC5A is a receptor, and we know that the mechanotransduction starts at the plasma membrane. We next confirm that GPRC5 expression is correlated to the stiffness changes. So to do that, we culture the keratinocyte on the different hydrogel, and we replate them on the glass condition. We observe that GPRC5A expression was always overexpressed during the transition from the hydrogel, so the soft, medium, rigid, to the glass surface. To rule out the GPRC5A expression due to the replating process, we include the glass-to-glass, and in which no significant expression was observed. We can note that the more the difference between initial and final substrate, the stronger the expression of GPRC5A. We also demonstrated with this result that GPRC5A expression is correlated to the stiffness changes. To illustrate these stiffness changes, we focus on the wound healing context, where we know that we observe an increase of the stiffness. We analyze the expression of GPRC5A in the wounded skin of mouse model. The immunostaining revealed that a specific expression of GPRC5A at the leading edge, the place of keratinocyte migration. We also observe that when we look in the region far from the margin and in homeostasis condition, GPRC5A is not detectable. This observation led us to study the role of GPRC5A in different cellular processes, such as the adhesion and the migration process, which is the main process that occurs during the repetitization phase. We analyzed GPRC5A relocalization during the early phase of adhesion, proliferation, and the differentiation. Our studies show a diffuse expression of GPRC5A when the cell is in suspension, an extreme soft condition. When the cells were plated and adhere in the surface, we observe a part of GPRC5A is translocated to the nucleus during the first phase of adhesion and during the proliferation. At the differentiation state, GPRC5A expression in the nucleus return to the cytoplasm. Making of this result, regarding of the result, we also would like to, identify in which compartment GPRC5A is expressed. Regarding to the nucleus, we show that GPRC5A colocalizes with the nucleolin, which is a specific nucleolar protein. Concerning the cytoplasm expression of GPRC5A, our immunostaining revealed that GPRC5A colocalizes with the endoplasmic reticulum with the vesicular trafficking, but not in the Golgi apparatus. This shows that GPRC5A translocation in the nucleus is correlated to the early adhesion process. To explore further the adhesion process, we represent GPRC5A expression using the siRNA approach. First of all, we verify that GPRC5A expression is repressed by qPCR and Western blotting. The analysis of the adhesion assay revealed that the assay GPRC5A transfected cells show a better adhesion compared to the control. We also analyze keratinocyte migration, and we show that the assay GPRC5A transfected cells delay the closure compared to the control. This result proves the causality between GPRC5 expression and the adhesion and migration process. We also analyze keratinocyte differentiation process. A limitation of the siRNA approach is the fact that they are transient. To get rid of this problem and to study GPRC5A involvement and the process and the long process such as the differentiation, we establish a stable cell line of immortalized keratinocyte entered, in which the expression of GPRC5A is silenced by shRNA. Of note, we have validated all of the previous results regarding to the adhesion and the migration process on the entered cell line. Our analysis of keratinocyte differentiation process in the 2D models show that the GPRC5A inhibition exacerbates the expression of marker differentiation such as the keratin 10, the involucrin, and loecrine. To go further in keratinocyte differentiation, we took advantage of 3D model of reconstructed epidermis. First of all, we analyzed GPRC5A expression during epidermis formation. The immunostaining revealed that GPRC5A is expressed during the first week and after is decreased during the stratification. This transient expression of GPRC5A supposes a role of this protein during the first step of keratinocyte differentiation. To test this hypothesis, we perform a reconstructed epidermis with SH-GPRC5A, and we analyze epidermis morphology at day 6. As you can see, with the hematoxylin and eosin staining, we show that in the SH-GPRC5A condition, we can observe two different areas. The first areas, we show that the thickness of the epidermis is decreased compared to the control. And in the second areas, we observe a disorganized epidermis with a detachment of the suprabasal layers. When we analyze a junction and the junction protein and keratinocyte differentiation markers protein, this immunostaining confirmed that the differentiation process is impaired by strongly reduced of eucadherin, as you can see here in green, and keratin 10, and also a discontinued expression of the involucrin in this condition. Moreover, the tunnel assay revealed a high proportion of apoptotic cells in the disorganized epidermis. To conclude, we demonstrated that in homeostasis, GPRC5A is not expressed. However, a disturbance of this homeostasis during the wound healing, in which the dermis mechanical properties is increased, this increase induces expression of GPRC5A, which may in turn increase the capacity of keratinocytes to migrate, reduce the cell adhesion in order to restore a functional epidermis. We also demonstrated that GPRC5A inhibition in the stiff condition show an alteration of keratinocyte differentiation, process, and this could lead to a disorganized epidermis. We can suppose that when the dermis mechanical properties is abnormal, like a soft substrate, the GPRC5A expression is reduced, and this can contribute to a disorganized phenotype. Finally, we can also hypothesize that in the soft substrate, where GPRC5A is reduced, GPRC5A rescue could lead to restore the epidermis functionality. In light of this result, we think that GPRC5A may be a key mechanotransducer, helping the keratinocyte to adapt to mechanical changes in different physio and pathological contexts. Finally, I would like to thank my supervisor, Romain Debray, Bérangère Fromy, and Philippe Faure, and also to thank the FDTC team, and thank you for your attention.
Thank you, Choua. Do you have questions for Choua? Yes, over there, please.
I have two questions. This protein, GPRC5, is a GPCR, isn't it? Is a GPCR?
Yes, it's a GPCR.
So this is a transmembrane protein?
Yeah.
So I was just curious how these kind of proteins can reach the nucleolus. And the second question would be, do you know if there is any relationship between forming this focal adhesion and the activity of this GPCR, or vice versa? If they have something to do together, or it's totally unrelated?
Alors, concerning the first question, the localization of the, in fact, GPRC5A is a G protein coupled receptor. And we know that this type of receptor is located at the plasma membrane. But another studies reveal that show GPRC5A localization into the cytoplasm also. Concerning the nucleolus localization, we used two different antibodies to visualize GPRC5A. One target the N-terminus and the other target the C-terminus. And we show you here is an antibody against the C-terminal. And we think we have a cleavage of this part. And is a short C-terminal of GPRC5A translocate in the nucleus. And the second, can you repeat the second question, please?
No, you're right, it was not totally clear. Because when the cells are on a stiff substrate, they start to make this focal adhesion, and these focal adhesion are mechanotransducers. So I was just wondering whether activity of a GPCR signaling, whatever, and the force that can be transmitted to the cytoskeleton, if there is any relationship. For instance, if you, I don't know, knock down your GPCR, do you prevent the formation of focal adhesion or vice versa?
We don't see the focal adhesion after the inhibition. We just analyze the capacity to form a precoce, the early adhesion. So I don't know if I answer your question. Yes.
Next question, please. Anybody would like to comment or ask something? Well, do you consider the bottom of the wound, any wound, to be a stiff substrate? I mean, you suggest that the keratinocytes would colonize the wound bottom.
Yes.
The bottom of the wound would vary according to the clinics, whether you just remove epidermis, whether it's farther deeper in the dermis. So probably the stiffness of this substrate for keratinocyte colonization would also change. How do you position your model towards this clinical reality? I mean, how to put this different way? You suppose that the keratinocytes would spread on the rigid surface, is it?
Yes.
But this rigidity varies according to the clinical situation. I mean, sometimes it's only just epidermis that is removed, and you have still basement membrane, for example. When the wounding is deeper, the mechanical properties of the wound, of the bottom of the wound, would change. So do you have any idea how the modification of the wound, epithelial wound healing would be affected by those mechanical differences between different wounds? No, not yet.
I think no, but if you want, we can discuss after, because I.
One-to-one.
Yeah.
Face-to-face, you know.
Just face-to-face.
Well, okay. That was the first step of your study, and then you can develop on this. Okay. Any questions?
Yes.
Yes, Lyon.
Only one. So, thank you for your talk. So, great hypothesis. So, just to know if you plan to check something in differential display between chronic wounds and acute wounds, to know if this kind of process is present or not in chronic disease without
I'm sorry, can you just speak a little bit higher?
Okay,
yeah.
I will do. I understand why you mentioned to speak face-to-face. So I just wondering if you plan to check in the lab of yourself the difference between expression or these global fields in chronic versus acute. Because in some chronic wound disorders, there is nothing that could be control the wound healing process. And do you think that this kind of target could be involved or not in this way?
for us, it's difficult to check this expression in another wound healing
Chronicle, long-lasting.
Yes, because we don't have the sample.
But you should get it. And you can do.
Leg ulcers are the chronic wounds.
Yeah.
So it could be one of the possibilities to study.
Okay.
Could be. But I let you engage talk with. Sorry, but so it's really nice. Great.
Okay. No more questions? Okay, no more answers. Thank you very much.